All right, thank you, Rizal, for the kind introduction. Yeah, even though I didn't age, I definitely gained some weight over the years. My wife always complains about it. When I met her 20 plus years ago, I was a lot skinnier and I actually have gained weight. So even though I didn't look like I aged, but I definitely changed. Anyways, delighted to be here. I was here 7 years ago giving a seminar in biophysics. I know the audience here has a lot of physicists. If you have questions, raise your hand and ask me. I'm going to try to give a rather general introduction so that I can set the stage. Maybe half of these terms that you see right now are unfamiliar to you, but hopefully as I go through the presentation, you'll understand what I mean by mechanobiology, mechanotransduction, and most importantly, cell skeleton, okay? So I want to start by motivating my interest in mechanotransduction. So I'm fascinated by the ability for organisms to sense and respond to forces. What you're seeing here is a few videos of C. elegans, a worm that in this video, when you touch it mechanically, it reverses direction. There are a couple examples in the biological, in the plant world, mimosa plants and Venus flytrap. When you touch it, it reacts mechanically. And then on the bottom right, I have a video of a dancing squid, and this is dead, but it still respond to mechanical touch. In this case, is actually due to the salinity of the soy sauce that actually triggers ion channels. But in any case, the squid is moving, even though it is actually not alive. So in any case, I'm fascinated by the ability for organism to sense mechanical forces and respond to it, okay? Now, I want to show a couple videos that are very similar in our field. This is cell migration. What you're looking at here is a neutrophil. So this is a type of white blood cell that's in your body. And neutrophils are tasked to essentially ingest bacterium. So here is a little bacterium. Surrounding this neutrophil are red blood cells. So this neutrophil is basically going to chase after this bacterium and essentially pushing away the red blood cells and eventually eats it. Okay, the ability for the cell to migrate is something of great interest to me. Is how does a single cell generate forces, be able to turn, be able to sense, and be able to eventually ingest this bacterium, as its food, right? It's not recognizing the red blood cell around it. So the cells are able to do it because there is a cell skeleton. There's a polymer underneath the membrane that's actually polymerizing, driving the forces that allow the cell to move. So we'll talk a bit about this today. So broadly speaking, I'm interested in understanding how a cell is able to generate force, sense force, and respond to those forces. And we do so by looking at single cells, collective cells, as well as building a system, a cell-like system that can do this. And I call this synthetic cells. And you'll hear a little bit about this later on as well. So our overarching goal, like I said, is how the cells sense and respond. And importantly is how the cells generate mechanical forces to enable their movement and then allow the cell to change shape. Okay. So we do this both in living cells as well as the cell-like system. I'm breaking down my talk into three little vignettes. The first vignette is a little bit longer. This is on matrix. This matrix is referring to the extracellular matrix that are surrounding cells. How did this matrix confinement modulate 3D spheroids, which are clusters of cells, and how do they allow these 3D spheroids to sort and undergo collective migration? So you'll hear background about this. And then I have two little vignettes on the synthetic cell system. First on reconstituting different type of actin networks in a giant vesicle. So this is our platform for synthetic cell. And then I'll talk about how this actin network architecture will govern the stiffness of this giant vesicle as a synthetic cell. And I'll close with a little bit of fun slides that I got to hear from Rizal yesterday. You'll see what I'm going to talk about a little bit. Okay, so just to set the stage a little bit more, this is an image of a cell or cartoon of a cell. And what is mechanotransduction? Really is the idea that mechanical forces can drive biological functions, and this is mediated through proteins or protein complexes at the membrane. They're called mechanosensors. Okay. So the physical stimuli are shown in blue. So they can be shear stresses, as in blood flow in the blood vessel that your endothelial cells sense. They can be extracellular matrix rigidity. They can be strain forces. Your cells actually can stretch. Imagine your blood, your lung and your bladder actually stretches, and this is physical forces that cells will sense. And then the cells ultimately respond with certain type of cell function. It can be changes in gene expression that happen at the nucleus level. It can also be changes in cell dynamics like migration. As I said, the sensors of the mechanical forces are called mechanosensors. They're highlighted in red. They can be mechanosensitive ion channel, so channels that senses force on the membrane, tension on the membrane. They can be adhesion molecules that link the cells together. They can be focal adhesions, which are protein complexes that are attachment points of the cells in the matrix, receptors, and certainly the cell skeleton, which are the polymers, the biopolymers that link the cell membrane to the nucleus, and certainly plays a lot of roles in cell rigidity and also cell dynamics. So my lab studied many aspects of this mechanotransduction. I'm not going to talk about all of them, but I'll highlight a few things that we work in this area. Okay. So what is actin? This is going to be, it's a textile model. I just want to introduce this to the audience here. So it exists as a monomer. It's a polymer that can polymerize from a monomer to a filament. And these are the construction material of a cell. Now, these filaments can bind to different type of actin binding proteins. And these proteins can essentially decorate on various parts of the filaments and regulate their dynamics or architecture. For instance, they can be proteins that bundle these, so basically linking them together into thicker bundles, and they're found in projections like filopodia. They can be motor proteins that slide the filaments together, like what you'll find in your muscle. There are proteins that will bind to the side. There are proteins that will bind to the end. This is called capping protein. The protein that will cross-link this filament together in a cross-link network. There are proteins that will sever the filament into two pieces. There are proteins that will nucleate the assembly of these proteins. So, given this diversity of actin binding proteins, you can imagine actin is a material that can rapidly remodel based on the cell demand of a cell. So relevant to this talk are two types of actin network that I want to introduce. One is called limitopodia. These are cross-link network, dendritic network that are formed by branches. It's branched by this protein called RP3 complex. Okay, and there's another type of network that are found at the leading edge of a cell. These are bundle network. They're called filopodia, and they're these parallel filaments are cross-linked together by a protein called fascin. Again, you'll hear about this. So there are different type of actin network, many of them, but the ones that are relevant to today's talk are this limitopodia, dendritic network, and these bundle network filopodia. So I wanted to show you another interesting features of a cell skeleton, which is the ability for it to self-organize itself. And the movie I'm going to show is a fascinating one. This I'm not sure how many of you have seen this before, but you're essentially going to look at a piece of a cell. It's coming from a fish keratocyte. So this is a cell that moves very rapidly. They're coming from the scale of a fish. And what this video is going to show is a piece of a cell that is actually doesn't have the nucleus, doesn't have any organelles, but when you mechanically touch it, so let me just pause it for a second. Okay, yeah. So you see, mechanically touch it, it breaks symmetry, and this piece of a cell spontaneously migrate, okay, in a particular direction here. So when I saw this video, when I started as a graduate student, I found it very fascinating because it tells you that the ability for a cell to move is entirely encoded by the proteins that are within this piece of a cell. It doesn't have nucleus, it doesn't have any genetic information, it doesn't have any organelles, but it has all the proteins necessary to generate self-movement, okay? So if we bring it back to the video I showed you earlier with the neutrophil moving to chase a bacterium, essentially the cells is able to sense a signal. And let's say if it's moving to the left, it's able to disassemble the cell skeleton and reassemble the cell skeleton in the new location at the leading edge. So allow the cells to navigate, to start moving. And this is how the cell does it at a very regular, the senses and respond all the time, okay? So I find this process fascinating. All right, so I'm gonna get into the first story, which is on this matrix confinement, how this regulate migration in a 3D environment. The context of this is collective migrations. I'm just showing you a few videos that you can readily find online on collective movement of organisms. So here, a flock of birds, they never collide. Here, swarming bacteria. I was talking this morning about swarming. These are, I don't know exactly what bacteria it is, but you have these very beautiful swarming behavior. And here you have MDCK cells. So these are epithelial cells moving on a pattern, adhesive pattern that you can see these cells are moving to the left, and they're not jammed. There's still movement that happens at all these three instances. So The questions that we're interested in is this jamming on jamming transition, and I like this quote from Liu Colloquium, a mathematical biologist, that talk about a living system happening in the edge of chaos where natural selection achieves and sustains such poised state. Um, I didn't show a phase diagram, but for those of you that know granular material, it can jam under certain conditions where you have high density, let's say low temperature and very little load, but you can unjam this by applying a force. And you can actually apply the same concept to a cellular system, which basically is what I'm going to talk about, is that we can unjam a cellular system by applying load to the cell system. So here are examples that people have already done using various infections on the 2D monolayer of cells. If you infect them with viruses, you can see this unjamming transitions that can occur in development, cancer invasion, and thank you, Rizal. That you can see this movement. And then here, similarly, this is in asthmatic cells, epithelial cells. When you compress the cells, you can start to see movement of the cells. Okay. So my lab is interested in looking at how mechanical perturbations of cells, in the case of 2D and 3D, that can actually start to stimulate movement of cells. The context of this is in metastasis in cancer. So here in cancer, what you have is a primary tumor that have cells that will basically escape out of the tumor environment, enter the bloodstream, and then exit out of the blood, and then start with a secondary site. Now, this is a very mechanical process because cells have to fight through all these extracellular matrix, be able to get out of the tumor. So I'm interested in looking at how does a cell escape from this primary tumor, okay? And this, and how does the confinement of the matrix influence this process? So cells are not in 2Ds, and many people have studied cells in 2D. When we first started this project, we studied this in 2D, but the story I'm going to present to you is the work that's done in 3D. So cells are here shown in a 3D hydrogel culture that consists of collagen, which is a very prominent or natural abundant extracellular matrix, and then alginate. And alginate is a hydrogel that can be cross-linked with calcium. So here is the experimental system we use is a collagen alginate composite hydrogel, and we can use calcium ion to chelate the alginate, and so allow the stiffness, the confinement stress to increase. And then when we add EGTA, we can lower the stiffness. Okay, so this is a very, very simple system that allows us to increase the confinement stress, look at what the cells are doing, and then we can reduce the confinement stress. Okay, so the question we want to ask is, do these changes in matrix stiffness govern collective cell migration? How do they impact cell migration? So there's one concept I'm going to use that's you're going to see later on, is this idea of differential adhesion hypothesis. So if you take two materials and they have different adhesion energies, in the high adhesion energies, with high cortical tensions or surface tension will tend to aggregate together with a lower surface tension to be on the outside. So this is what you will happen. You'll see this in tissues as well. So let me just show you this. So this is what's being proposed to allow tissue sorting, where the high surface tension cell types will be in the lumen, and where the low surface tension cell type will be at the peripheral. Okay, so this is called differential adhesion hypothesis. So the question then is, can we replicate this? A lot of groups have replicated this, but if you start with a sorted tissue, the idea here is that maybe the lower surface tension cells will be able to escape easier out of the matrix. So for this, because of the cancer context, we start out with we're going to work with 3 cell types. Given the audience, let me just walk you through the cell types. The name itself doesn't matter, so we work with a normal cell type. This is called MCF10A. And then we work with 2 cancer cells. One is called MCF7. The other one is called MDA, MD231. They increase metastatic potential, so the ability to leave the tumors. Okay, so we do a co-culture, which means that we mix the 2 cell type together. We generate this spherical cell aggregate. So this is what we call spheroids. And you can see this spheroids consists of 2 different 10A cells. We label them with 2 different markers, one with EGFP, one with RFP. And this is over time. You don't really see much of a difference. Now, if you co-culture the 10A cells with the MDA231 cells, you can see the green cells are more migratory. They're a little bit, they have greater movements, okay? So we can analyze this by looking at their mean square displacement and as a function of the confinement stress, okay? And then we can calculate, or we can essentially get the exponent out of this relationship, and you can see that the higher the number, if it's 1, it's diffusive. If it's greater than 1, it's even more, I guess, more migratory. And this happens in the low calcium chloride condition compared to the high calcium chloride condition, where it's basically confined. Now, the 10A cells are less migratory compared to the MDA231 cell, which are what we expect because the cancer cells are just moving more. Okay, and we can analyze this by looking at the effective diffusion coefficients, and as you can see, the MCA10A is less migratory, less diffusive, but it lowers diffusion when you have higher matrix confinement, right, at the 10 millimolar calcium chloride. So this all makes sense. That the fact is that if you apply compressive stress or confinement stress here, that the cells are just less motile. Okay. Now, what happens if you introduces sorting behavior, and we're not really seeing sorting here. And how can we enable sorting? So sorting here, other people have described, migrations in a single cell versus collected cell. And there's this paper from some years ago that shows that when the collagen density increases, and you can actually get single cell to leave this matrix, whereas if you have low collagen density, then it will have more the collective migration happens more in the higher density, whereas here in the low collagen density, you have more single cell migration. So there's a switch between single cell moving versus collective cell migration. Now, sorry, this gets a little bit deeper here. Our experimental system uses a constant level of collagen, whereas we vary the alginate cross-linking to obtain different confinement stress. So even though there's a study previously describing collective versus single cell migration out of these spheroids, our experimental system is actually different in that regard because we have the same collagen density. Now, when we switch over to so this is the experiment showing the 10A cells and 231 cells as functions of time. In day 0, we have a very well mixed spheroids, and you can see that when you have very low collagen, very low calcium concentration, the single cells are moving away. And then when you have high collagen, then they're actually less migratory, again, as you expect. So we can calculate, so this is a cell aspect ratio. The aspect ratio describes the elongations of a cell. So the cells are very elongated. They're more migratory, and they're more migratory at the 0 millimolar calcium level, and it decreases over with calcium. We can also describe the distance index. This is describing the location of the cell relative to the radius of the spheroid. So if the distance index is 1, basically the cells are at the periphery. If they're greater than 1, they have escaped out of the spheroids. And you can see that when you are at 0 millimolar calcium, it's greater than 1, whereas at 10 millimolar, they're basically trapped within the boundaries of the spheroid. Again, it makes a lot of sense. So this is metastatic potential actually influences migration behavior under confinement is sort of the question I want to address here. Now, we can't really do this with MDA231. So we went back to the 10A cells and asked, so this is with MCF7. So MCF7, remember, is a less metastatic cells. So the 231s, escape out of the spheroids. Now, interestingly, when we switch the 231 cells with the MCF7 cells, we start to observe this phenomenon of sorting. That you can see a cyan cell surrounding the magenta cells. Now, this started to behave like the sorting pattern I talked to you about. The MCF7 cell could be less adhesive compared to the 10A cells, which leads to the sorting. And this happens only in the scenario where you have high calcium chloride. And to this day, we still don't exactly understand why. But you can see from the analysis here, looking at this index, that at the 0 millimolar calcium chloride, these two cell types have roughly the same, they're reasonably well mixed. But when you go to the higher confinement stress, you start to get the sorting, where the MCF7 is at the periphery of the spheroids, and then the 10A cells are compacted in the middle. Um, and this correlated with the adhesions. So what I did not introduce to you is this molecule called E-coherin, and this is the molecule that allows cells to bind to one another. And in the case of 10 millimolar calcium, you can see the red is very pronounced in the center, and then less in the periphery. So you can start to see this differential expressions of this molecule E-coherent on the cells. Again, supporting the fact that you have differential adhesion. Now, how can we manipulate this in a living system? So does the cell sorting depend on E-coherent expression? The answer is yes, because we know from before this differential adhesion hypothesis. But here we can experimentally test this, is that these are our control cells, and they look exactly the same when we use MCF10A. They have the same adhesion. They're just the same cell type with two different color. And you can see that they don't sort at all, as you would expect. Now, if we take a knockdown approach, we can use a genetic approach to knock down E-coherin. So here is a Western blot showing that the E-coherin is completely knocked down. Then we can recapitulate this sorting behavior, that at the high calcium concentration, we actually see this behavior where the low adhesive cell, which are the one in cyan, is on the outside, and the high adhesive cell is on the inside. And this is the same cell type. Okay, just they're the same identical cell type, but with different adhesion properties. And again, we can quantify this. So all this stuff so far is actually rather, I would say, expected, but we wanted to understand what happens if you reduce the matrix stiffness. We have this hydrogel system that allows us to increase the stiffness, and we can reduce the stiffness, so we can add EGTA to it. So the experiment is as follows: that we added calcium chloride to crossing the alginate day 0. We wait for 4 days to allow sorting to happen. We add EGTA to crossing the alginate. So here, this is our control, where again you have the two MCF10A cells. The cells remain well mixed. Now, if you then treat the cells with EGTA, you can see this behavior or phenotype where the cells are no longer compact as a spheroid, but now the cells have escape, right? And this is a sorted pattern, right? So you have the cyan cells of the periphery, the magenta cells of lumen, but in the presence of EGTA, the cells are no longer compact as a spheroid. So you see the escape of these magenta cells, and certainly the cyan cells as well. So this is rather intriguing that we have actually somehow break open the spheroids. I think this is my favorite movies, and you can see in a second, this is our control. When you don't add EGTA, okay, these are cells that have, sorted, and essentially the idea here is we have very high internal pressure that are generated by this confinement, and then it's balanced by this external pressure that's provided by the extracellular matrix. But when we add EGTA, look at, this is the same experimental conditions. The spheroid just bursted, and you have all the cells that escape out of the spheroids. And this is just a really fun movie to watch. And there's a lot of, I guess, I was surprised to see there's many cells there. This is over 24 hours. So we have high internal pressure, but when we lower the pressure from the outside, then the cells have basically escape out. This depends on sorting, right? So this is sort of a montage of time-lapse images, and we can track the cells, and they're basically leaving out of this potentially on these collagen fibers. But like I said, this depends on sorting, and we can do the experiment when we don't have sorting, right? So this is MCF10A cells again, without knocking down the E-coherin, and you don't have a sorted state. This is done under high calcium condition where you have high confinement stress, but when you add EGTA, there's no unjamming happen here. So you can see the analysis here where when you have a knockdown in this blue and green, the green bars are showing that you actually do have this unjamming happen. Again, only when the cells are, when these spheroids are allowed to undergo this sorting process. Okay, yeah, and this sort of supports the motility is slightly increased, but not a great amount. So we also collaborated did a collaborations with a modeler who essentially came out with this SPV models as a Verneuil model where they can actually model the adhesion between cells. So there is two types of cells, A and B, and then there's a matrix. You don't see the matrix, but there's also adhesion energy between the matrix. After some amount of time, given these different adhesion energies, they can certainly sort. This is not surprising. But they also were able to model the confinement stress and show that, let me just bring this up. And when you have a high compression, it would basically have a sorted state. When you have medium compression, you sort of have cell escape. And in the case of reducing the compression, you have essentially cell can disperse out of this sorted spheroids case. So again, similar to what we find. It's not surprising because we have essentially reduced the external pressure, and now the cells are able to escape. So by downregulating this confinement, we find that the cells can actually disperse into the extracellular matrix and under only sorted conditions. Okay, so this is our final model that essentially we start with a uniform spheroids and allow the stiffness dependent sorting to happen, and we still don't know exactly why that happens, but under this high calcium conditions with crossing alginate, we generate the sorted spheroids, and then the sorted spheroids can escape, can invade once you essentially reduce the confinement stress. Okay. So I'm going to switch gear a little bit to talk about our in vitro experiments. So the next two short stories about our in vitro experiments, and this is work done by Yashar, a former postdoc, on how we can study self-organization within the confined system and looking at actin. So about 2/3 of my lab work in this area called synthetic cells, and this is probably very new to you. The way we think about synthetic cell is a cell-like system that has a lipid bilayer encapsulating molecules of interest. Our lab specializes in expressing membrane proteins on the surface of a synthetic cell, but we can also encapsulate any type of biomolecules we want inside this synthetic system. The way I want to I think about synthetic cells is, fascinated by how cells can take input and produce an output. We want to be able to build a cell-like system that can take, sense any type of inputs, forces, molecules, or light, and then produce different type of outputs. My lab has looked at mechanosensitive channel, other channels of interest, but today I'm going to talk about just things that are encapsulated inside cell skeleton, encapsulated inside that can generate forces and change the shape of this synthetic cell. Okay. The kind of idea or overarching goal is to assemble biological components in a meaningful way to build an artificial cell that will mimic certain aspect of a cell. So people call this artificial cells for synthetic cell. And depending on what process you want to emulate, you can build different type of synthetic cells. So here, the players are two cross-linking proteins. So one is called FASN. And this is a short cross-linker of 6 nanometer in size. And then we have a long cross-linker, alpha-actinin, that is 35 nanometer in size. They're found in different regions of a cell. The FASN is found in filopodia, and the alpha-actinin is found in these contractile ring during in cell division. So people have already done experiments with single filaments and looking at these two cross-linker, and there's a very simple model that can explain experimental observation, which is the fact that you find these domains in these two filament systems. So the long cross-linker would preferentially be on one side, and then you have the short cross-linker located in a different domain. The reason for that is you want to minimize bending energy, right? If you have two filaments together, you don't want to have them go in the wavy format. So instead, they will basically cluster all the short cross-linker together and have all the cross-linker on the other side. So, the question I want to ask is, does this hold true if you encapsulate this cell skeleton in a synthetic cell format? So the way we build these synthetic cells is a very simple way to do this. Is we essentially emulsify our protein of interest in a droplet, and then build a bilayer vesicle around it or bilayer membrane around it. So here, the process is very simple. Is we have an oil lipid mix and we have aqueous solutions. We just sort of pipette up and down or vortex it. You get these water and oil emulsion. They contain lipids, so there's a single layer of monolayer lipids, and then you put this in a little device, a 3D printing chamber, rotating, and we can basically get these droplets to fly through this oil-water interface and generate another lipid monolayer, so you have a lipid bilayer. Okay, and here's examples of what that looks like when we use actin and another cross-linker. So you don't see the circle, you don't see the membrane, but you see these objects are basically encapsulated within lipid bilayer. And we call this giant uvular vesicle, or GUVs. They're heterogeneous in sizes, but they're very, very easy to do. Very quick, relatively simple to do. And then we can image them right away on a 96-well plate. Okay, so we first started out with doing this experiment with fasten, this cross-linker, the short crossing I talk about, because they can bundle. These filaments will basically form a protrusive filament or bundle of filaments, and this is a very beautiful, demonstration that actin can generate enough force to form a membrane. Right, these are bundle actin filaments that literally pushes out of the membrane, and much like a stress fiber. And we can use light to depolarize to break down these actin, and over time you can see that these actin depolymerizes and the vesicle becomes spherical because the filaments are generating forces to push out against the membrane, but they can they support mechanically support the shape of the vesicle, right? So here they depolymerize over time. So what happens if you add a second cross-linker, alpha-actin, the long cross-linker? So if you add the cross-linker, if you just use the alpha-actin alone, you see something very different, right? So instead of these bundle actin, you see a variety of actin architecture. So you have, and that's size dependent. So depending on the size of the GUV, you can have things that are like a ring when you are a small GUV. And when you have a large vesicle, you have network. And it changes as you increase in the alpha actin to actin ratio. So you start to have various morphology when you start to incorporate, a single actin binding proteins. Now, if you mix the two together, which is the interest that we have, is you start to see another phenotype, which is an aster phenotype. So here, just give you an example, these are field of view of multiple, but you start to see a region where you have very dense actin in the middle, in the center, and you have these bundle that radiate out from this center aster. Now, this sort of remind you of the differential binding of crosslinker on actin. And when we do this with multiple labels of protein, you can see that the alpha actinin, the long crosslinker, is preferentially localized in the center. So we can do a line scan across, and you can see the alpha actinin is enriched, and fasten is not really there. If you do a radial line scan, and this is with label fasten, you see the little spikes of fasten that shows up because the fasten is the bundle that radiates out of the center. The alpha actin is in the middle, actin is everywhere. So you have this, self-assembly process of two actin binding proteins along with actin. And we collaborate with Aaron Dinner at University of Chicago, and he did some coarse grain simulation here with alpha actinin or fasten alone, and these are probably density map. They don't look very different, but if you combine, having alpha actin and fasten together in the same simulations, you will actually see alpha actin is enriched in the middle. It's really hard to see from this image, but they're, I would say, more depleted in the center, and there's more fasten in the outside of the center region. So kind of coincide with our experimental observation, where the center of the aster is enriched with alpha actinin, and these radial bundles that come out are preferentially fasten bundles. So this is a complete self-assembly process. Let me also emphasize that this only happens if you have confinement. If we take the same three proteins and we put it in a solution, they don't generate these aster geometry. So you have to put them in a vesicle where there's membrane interactions with the cell skeleton. Then you have this behavior. Okay. So let me quickly summarize this part that we've looked at just fasten alpha actin, but the same kind of competition sorting behavior, maybe not sorting, competition behavior happens for other actin binding proteins as well. So in my lab, we've looked at this RP3 complex and fastened together, and this was a paper that we had a few years ago. But I think it's generic enough to think about how different actin binding proteins can compete with one another in a cell. So here in this particular part, I talk about how crosslinker can self-assemble into sort of distinct domains in a GV format. Okay, so let me transition to the third vignette, which is rather short. And this is done by Nadab, who graduated a year ago and now a postdoc at Harvard. And he was interested in looking at how these different type of actin network can govern the mechanical properties of these giant vesicles. Okay. So here, there's a bunch of different techniques one can use to characterize mechanical properties of cells or giant vesicles, including micropipette aspirations, AFM, atomic force microscopy, acoustic radiation, electric deformations, or some type of optical trap setup. So that, in his dissertation, worked with a couple of these, including pipette aspirations and electric deformations. I'm going to briefly highlight some of his findings. So he set up this electric deformation, which is apply a alternating electric field. This is just using two copper tape as electrodes, and the idea here is that you can generate a shape change based on applying this electric field, and we can measure the long axis and short axis, and just essentially get the aspect ratios of a GUV of a giant vesicle. And then idea here is that we will encapsulate different type of actin and look at how this actin will alter the mechanical response when it's subjected to this alternating electric field. So in the case of adding just buffer alone, and this is a very transient, so we only apply this for a few seconds. The aspect ratio is 1.45, so you can see the GUV certainly deform into this ellipsoid and revert back to a spherical shape. Now, if we encapsulate filamentous actin, okay, this is 5 micromolar filamentous actin, we dampen the shape deformation to 1.23. The vesicle is less deformed, as you would imagine. There's a viscous material, there's polymer in there. So what happens if you take the same polymer and arrange them differently? So and maybe this is not too surprising. We have ways in our lab to nucleate this actin at the membrane, and I didn't go into the biochemistry of this, but this is called a cortex. And this is actually what happens in cells. The cell is actually not that fragile. It's able to move because of this cortical actin is dynamically remodeling the cell membrane. And so we can attach in some way of this actin in the cortex. And you can see that the vesicle barely change shape, right? I mean, there's some observable changes, about 5%. And we can also cross-link this actin using alpha-actinin that I introduced earlier. So we can take the same amount of actin and we can cross-link them, and they will also not change shape very much. So in essence, the simple concept here is that depending on how you arrange the actin, it's the same amount of material. It's the same amount of filamentous actin, but we arrange in different fashion, and then you can get them to mechanically, I say resist the deformation, right? So the cortex is 1.05, and here is about 1.12 or something like that. Okay, so this is a very simple illustration that actin is providing the mechanical support of a cell. Now, Nadab also attempted to look at how applying a mechanical force will alter cell skeleton dynamics or cell skeleton structure. So here, what you're looking at is a set of pipette aspiration. So this is a micropipette that we can apply negative pressure and aspirate, suck on a vesicle. And so here, the idea is that if we have bundles of actin filaments, we can apply a local force, using the pipette, and we can see how this bundle filament rearrange. We can also take the same bundle and cross-link them using a protein, in this case, RP3 complex, and see how they respond. So I'm only showing one example here. So you can see here that when we have these bundle filaments and we apply a aspiration, you can see the pipette is not visible here, but we're showing, you can see how these bundle filaments collapse into a thick bundle. So if you imagine a bunch of rods, and if you apply a point force, these rods can align themselves, almost like thinking about locks in a river. If you have the river flow, the rods or the wood, I guess they will basically align. The locks will align themselves into the directions of the river. Now, where if you have branch bundles, because they're cross-linked together, they're actually much more resistance to the applied forces, right? So these bundle filaments or bundle branch bundles barely move or barely reshape under this applied load, and again make a lot of physical sense, right? So they're not able to move. So in some sense, you can think about this as what happened in cells. The cells, when the cells need to move, they need to reorganize their actin. They need to restructure their cell skeleton in the direction of movement. If they're very cross-linked, they're actually unable to do that. Whereas if they're more less cross-linked, they can actually remodel. Okay, so let me quickly summarize this. We're doing well in time. The last part is I kind of summarize it in this graphical things. Many people have know that if you build a truss-like structure, they can support load, and this is a toothpicks. and they can support cinder blocks. I mean, pretty impressive. But you can also have toothpicks that completely falter if you don't know how to build a proper structure. So the idea here is that the architecture of a cell skeleton really matters, right? I emphasized earlier that these encapsulated systems that we use the same amount of actin. They're all 5 micromolar, but depending on how you organize these filaments, they can resist load differently, right? So the case that we resist load the best is when you have a cortex, but they can remodel into crossing, or they can remodel as a filamentous actin, and they have differential resistance towards this load. Okay. So the architecture matters, and they're really important for deformation resistance. Okay, so that completes my sort of scientific portion of the talk. But Rizal sent me a text message yesterday and reminding me that I can talk about something slightly off today's topic, which is on the mechanical biology of microgravity. So I only have a few slides here, and I'm not going to go over any data. The short of this is that my lab received a grant a few years ago to do experiments on the International Space Station. So I'm going to just tell you a little bit about that experience, but not on the data itself, because it's a little bit too dense to share that in today's seminar. But the question is, I think it relates well to the topic today, which is on cell skeleton in general. I mean, we all sense and respond to gravity, but when you are in space, the cell skeleton would certainly respond differently to the lack of mechanical forces, in this case, gravity. So this is the same diagram I had before. In this project, there are 4 students and postdoc that was involved in this project throughout the lifetime of this project, which is to see if we can understand the cell skeleton response on the ISS, and specifically on bone cells. So osteoporosis is a condition where bones become brittle, and astronauts have reported, or I guess people have reported, that astronauts going to space come back with brittle bones and weakened muscles. And this is a condition where it's actually hard to study on Earth unless you have a patient that does that. So the premise is that by doing experiment ISS, there's a accelerated environment where we can actually study a load limited conditions, right? So that was the whole premise, and that got us funded in 2019 from the International Space Station with NSF. And I know some of you know this. Mark Kelly is I guess local to Arizona. There was a paper published in 2019 that basically studied this twin astronaut study where his twin brother was up in the ISS. They came back and did a whole bunch of metabolic assistance biology measurements, and I only highlighted one. I don't think we need to read into this, but just the fact that the two, identical twins have different responses in their bone in general, and it's at different phases of how it is in flight or post-flight. So this got me interested in trying to understand if bone cells would respond differentially to Earth condition versus the ISS condition. Okay. So with that, I only have 2 slides. Again, because the science is a little bit more complicated, there's a biological hypothesis behind it. Just to say that we have to collaborate with a industry partner. This is a company based in Kentucky, Space Tangle, and they specialize in packaging your experiment for the ISS. We don't physically go up there. Astronaut times are very expensive. So their whole spiel is that they can automate your experiments. They put everything that you want to do in a tiny box, in a box like a shoebox size. So this is one of the experiments. We do 2 experiments, and this is a well plate, with spheroids in there. So the spheroids I talked about earlier was in there. And the first flight we did was with NG-18, and this is a fly out of Virginia on November 4th, 2022. It's a Friday at 6 p.m. We had to basically pack our experiment. We worked the whole day that day, and the thing that I guess the name of this product they do is called Cube Labs. So these Cube Labs would go into this PAL, which is these locker. So there are 2 experiments, one on top and one on the bottom. And you can kind of get a sense of their sizes. They're not that big. Okay, so this whole locker would then get loaded into the capsule. So launch itself was scrubbed 10 minutes before takeoff, and this is something that you kind of see on TV, but I experienced on live. I was sitting there, counting down with the rest of the folks, and then literally say, okay, launch a scrub because there was some fire alarm pull at a control center in DC. that basically tell them to stop. And it was just weird to sort of, it wasn't things on the ground at Virginia. But in any case, they rescheduled this for 24 hours later, and it was launched on November 7th. And real quick, it takes a while. It takes hours to fly across the country, but it takes a while to get to the ISS and certainly to dock and all that stuff. So the removal poll was sometime on Thursday, November 10th. Here's an astronaut flying with, or not flying, kind of gliding with our Paul locker, and then installing that. Throughout this time, this was not powered. There's an onboard battery that power this, and then it goes up, keeping it at 37, and so on and so forth. And then it gets connected to their power on the ISS. Long story short, this experiment failed. Because that experiment failed, I managed to get them to give us a second flight. And that was actually fun because at this time I also get to go. And this is at SpaceX 27. This is in Florida. And because of the scheduling, this was in March, on March 15. And I was at the Kennedy Space Center for about 2 minutes. Went there and get a badge. And they shuttle you from the Kennedy Space Center to a NASA site that's on site. So you need to have a badge to go in there. Because of the schedule, I think I had to teach that day. I took a flight. I got there at 5 p.m. I rented a car, drove there, got the badge, and then basically was taken to the secure site. And this is a building. Why is this special? This is where Artemis was built, put together. Apparently, this is the largest single building with the largest volume, I guess they call. It doesn't look that big, but it's humongous. And then this is the final slide I have. This is the launch itself. The launch was at 8:30. And because I have other meetings the next day, I basically was in Florida for less than, I don't know. I took the flight the next morning at 5 a.m. So after the launch, I drove back to Orlando Airport, stayed for a few hours, ate dinner at 11. Basically was at the airport at 4 a.m. On the way back. So let me just show you the, so it was fun to, I don't know if I could. Sorry, this. There's definitely my voice in there, but also my students. I brought 2 students with me. It's pretty cool. It's a short video. Someone's right. Yeah. So yeah, I don't think I'll ever publish in this journal again, but there it is. I have published in I guess MBJ microgravity. I've never heard of the journal before, but apparently it's a decent journal for this field. But anyways, the work has been published, so it's all good. All right. Lastly, thank all the folks who do the work. Actually, most of the things I presented today are recently published. The folks have actually left my lab. They're either postdocs or in other positions. I collaboration with Max P at Northeastern, Aaron Dinner, and then Space Tangle and the funding agencies. Thank you. That's all.